Now, this is a very high yielding table. It can look pretty crowded, but I shall help you to navigate through this. Now, if you look at the first box, that is toxigenicity and invasiveness. The only invasive diarrhogenic E. coli is enteroinvasive E. coli. All the others are non-invasive: enteropathogenic, toxigenic, hemorrhagic, enteroaggregative, and diffusely adherent. All of them are non-invasive. Then, which are the strains that produce toxin? So, the toxigenic ones are, enterotoxigenic is toxigenic definitely. Then, enterohemorrhagic produces a toxin, and also enteroaggregative, that is also producing a toxin. So, three toxin producing ones are enterotoxigenic, enterohemorrhagic, and enteroaggregative E. coli. And the only invasive one is enteroinvasive E. coli. So every time you think of enteroinvasive, you have to compare it with shigellosis. Okay. Now, the mechanism of action of enteropathogenic is attaches to the intestinal mucosa by the BFP, that is bundle forming pili, and forms a sort of a pedestal-like structure, and it produces something called attaching and effacing lesions. So that is what they had asked in the question. So it causes attaching and effacing lesions by means of BFP, bundle forming pili. Now, enterotoxigenic, we are all familiar because of the association with traveler's diarrhea, and it attaches to the intestinal mucosa by CFA, that is colonization factor antigens, and it produces toxins. So, there is labile toxin and stable toxin. So, LT goes and attaches to the GM1 ganglioside receptors, and it acts via increasing CAMP. So, what it does is it activates adenylate cyclase and thereby results in increased CAMP, and all the actions are because of this. Whereas a stable toxin activates guanyl cyclase, increase cGMP. So increase cAMP, increase cGMP, and attachment is by CFA. Okay. Then enteroinvasive, that is, the name has the word invasive. So the mechanism of action is invasion of intestinal mucosa by virulence marker antigens, that is VMA. Okay. So invasive action by means of VMA. Then comes enterohemorrhagic. It has got the virotoxin or the Shiga-like toxin, that is VT or SLT, that is the name of the toxin. It attaches to the globotriaoscyl ceramide receptor, that is a GB3 receptor, and inhibits protein synthesis. So VT or SLT attaching to GB3 and inhibiting protein synthesis. It has got a predilection for the endothelial cells that is responsible for the MAHA, this microangiopathic. So it can even produce microangiopathic hemolytic anemia. Then enteroaggregative. So it is aggregative. So it has got aggregative adherent fimbriae or AAF1 that causes colonization. And it produces a toxin, EAST1 toxin, and adheres to HEP2 cells in a stacked brick pattern. So it's got AAF1, that is an aggregative adherent fimbriae 1, brings about mainly colonization, and a toxin EAST1. And on HEP2, it forms a stacked brick pattern. Whereas diffusely adherent is colonization and diffuse adherence fimbriae 1. So DAF1, and it's got a diffuse pattern on HEP2. So if you look at it, EAEC and DAEC have some things in common and some things different. The main difference is that EAEC is toxigenic. It produces EAST1 toxin. Otherwise, it's got AAF1, the other one has got DAF1. Both cause colonization. One has got a stacked brick pattern on HEP2, the other one has got a diffuse pattern on HEP2. So now I hope this table is clear. So remember ETEC, labile toxin, stable toxin. Remember EIEC, invasive. And enterohemorrhagic, it's got virotoxin or Shiga-like toxin, protein synthesis inhibition, and microangiopathy. So EPEC mainly causes infantile diarrheal outbreaks, and it's mainly seen in the developing world. ETEC, we know it causes traveler's diarrhea as well as acute watery diarrhea in infants and adults. And the toxin production detection is by ELISA or using PCR gene to detect the labile toxin or stable toxin. So you can have ELISA method, or you can go for a polymerase chain reaction for detection. Enteroinvasive, I said you have to think about shigellosis. So it causes dysentery, just like bacillary dysentery, just like shigellosis, and it's very closely related to Shigella. and again seen in the developing world. And diagnosis is by Sereny test. Just remember this name test, it's not used now. Then HeLa cell invasion and also VMA ELISA. So virulence marker antigen ELISA, Sereny test and HeLa cell invasion. Whereas enterohemorrhagic, the name has got hemorrhagic in it. So it produces hemorrhagic colitis and hemolytic uremic syndrome. And one very important strain is the O157H7. And the food items contaminated include lettuce, undercooked beef, certain sprouts, and so on. And this is a disease that is mainly seen in the industrialized countries. So far, we were talking only about developing countries. And diagnosis is using a special media, that is sorbitol McConkey agar, in which it does not ferment sorbitol. Toxin detection methods are again ELISA. Then you can have cytotoxicity on Viro or HeLa cells, and PCR detection of the gene for virotoxin. So ELISA, PCR detection we have discussed even with respect to ETEC. Then there is a sorbitol McConkey agar and cytotoxicity on Viro or HeLa cells. Then comes entero-aggregative, that causes a persistent diarrhea. And here the strain responsible is O104H4. Okay, and that produced a Shiga toxin and even caused HUS. So that is one thing different from the usual entero-aggregative E. coli. This one has produced a Shiga toxin and even caused an HUS. Okay, and it's mainly seen in the developing world. So from this box, what you understand? One is traveler's diarrhea due to ETEC, infantile diarrheal outbreaks due to EPEC. Entero-aggregative produces mainly a persistent sort of diarrhea. For ETEC diagnosis as well as EHEC diagnosis, you've got ELISA and PCR. EHEC also you have sorbitol McConkey agar and you have the cytotoxicity on the Viro or HeLa cells. EHEC causes hemorrhagic colitis and hemolytic uremic syndrome, and it's a disease mainly of the industrialized countries. And then enteroinvasive, you have to always remember it is just like shigellosis. So you can it will produce dysentery. Sereny test can be used here. VMA ELISA and HeLa invasion can also be used to diagnose this.